This will serve as your pre-recorded or video lesson with regards to biochemistry. So we've already done discussing the two major macromolecules, and that was the carbohydrates and the protein. So now let's move forward to discuss another macromolecules that is very important to know in or to know for you to know. It's very important for you to know because that is one good components inside your body, and that what's the lipids, or also known as the fats. Literally, when we say lipids, this is from the figure below. It's called that fats and oils are the most widely occurring types of lipids. Thick layer of the fat help insulate polar bears against the effect of low temperature. So basically, fats and oils not only give a disadvantage to the body of a certain of a certain organism, but hence it also give importance, or it also protects you from a given situation. Like for example, in the picture below, that the polar bear, the fats and oil of polar bear is the one who help it to insulate against the effect of low temperature. That's why the polar bear is not usually can be found on those country that is very cold. Okay, so that makes the fats and oil different to the other macromolecules. Moving forward, when we say, so that's define how lipids as fats provider a major way of storing chemical energy and carbon atoms in the body. Fats also surround and insulate vital body organs, providing protection from mechanical shock and preventing excessive loss of heat energy. One good components of the lipids or the fats are phospholipids, glycolipids, and cholesterol. These are all components of a lipids. And another one is a several cholesterol derivatives function as a chemical messenger, like for example on hormones within the body, that also explains lipids. So that's why again, when we say literally lipids, it explains a fatty compounds. When we literally said fatty compounds, it actually perform a variety of function in your body. There can be part of cell membrane. That's why I'm saying the one good components of the lipids is the phospholipids, glycolipids, and cholesterol, because these are part of your cell structure, and that is called cell membrane. What does it help? It actually help to control what goes in and out of your cell. They also help with moving and storing energy. Not only that, lipids also absorb vitamins and making and hormones. So that explains how important lipids are inside your body. So literally that explains the definition of the lipids. So don't put in your mind that lipids only has a disadvantage in your body, but hence lipids also has a lipids can be a provider, can be a protection to what? To serve as a building blocks for all living cells. So that's how lipids occur. It regulates your hormones, it transmits your nerve impulses, and even store energy in your body. And how does the lipids can store energy? If the carbohydrates is a source of energy, lipids can store energy. Why? Because by the means of our body fat. So that explains what is lipid. Okay, so that explains the definition of lipids. So now let's move forward to the structure and classification of lipids. So right now we will not only be focusing on the definition of lipids, but hence we will be going to identify also the classification of it and the structure of it, starting with explaining that unlike carbohydrates, the most other classes of compounds are lipids. Lipids do not have a common structural features that serve as the basis for defining such compounds. Instead, their characterization is based on solubility characteristics. So when we say solubility, going back to your general chemistry, that explains actually the ability of a substance, or when we say substance, as we all know, in a solution there is a solute and solvent. When we say solute, this is the dissolving substance. And when we say solvent, this is the dissolving medium. When we say dissolving substance, these are the substances that to be dissolved, like for example, solid material. And when we say literally a solvent, these are the substance which dissolve the substance or the dissolving medium, and that explains the water. So that's why if we literally explains the structural form of lipids, it is not different, or it's not the same with carbohydrates, because in lipids we are not basing on the characterization of each structure. We will be basing on the characteristics of solubility, because it explains the nonpolar and polar structures. So as we all know, when we say literally lipids are fats, it has a components of water. Okay, so that explains this slide. And moving forward, lipid is an organic compound found in a living organism that is insoluble or only sparingly soluble in water, but soluble in non-polar organic solvents. When a biochemical material, like for example a human, animals, or even plant tissue, it is homogenized in a blender and mixed with non-polar organic solvents. So the substance that dissolve in the solvent are the lipids. So literally, lipids are insoluble. So what do you mean by insoluble? So literally, when we're trying to identify what is insoluble, this is basically an impossible to dissolve. But in the case of lipid, it can be sparingly soluble when you combine it in water, and but soluble in non-polar organic solvents. So literally, if we're trying to explain it, it can be it can be soluble if you combine it in water. But literally, when we say lipid, it is insoluble. It is non-polar organic solvent or non-polar organic substance. So that's why it explains that the substance that dissolve in the solvent are the lipids. Okay, so if we literally combine the lipids and the solvent, the lipids will dissolve. Okay, so this is the material, or the one good example of a solid material. Why? Because lipids can dissolve if you combine it on the solvent. Okay, so that explains this slide. Moving forward to another. So figure 19.1 shows the structural diversity that associated with lipids molecules. Some are esters, some are amides, and some are alcohols. Some are cyclic, and some are polycyclic. So the common thread that ties all the compounds of figure 19.1 together is solubility rather than the structure. So again, going back to what I've said a while ago, we will be focusing not only on the characterization of the structure, but hence we will be focusing also with the solubility characteristics. Why? Because in lipids, if we literally explains how the structure combine, compose, and connect, it can explain by its connection or attachment to solubility characteristics. So going back to that figure, so this explains the figure 19.1. So this is the constant formula for fat, biological wax, steroid, glycerophospholipids, sphingophospholipid, and sphingoglycolipid. So literally, it's constant. It's a good example of lipids. I want you to memorize all the structures because you know already that in the examination, structure is very important. Okay. Next, two common methods exist for subclassifying lipids onto families for the purposes of the study. One method uses the biochemical functions of a lipid as the basis for classification, and the other method is based on the whether or not a lipid can be broken down into smaller units through basic hydrolysis, that is reaction with water under basic conditions. So literally, since a while ago I've said that the lipids is insoluble but can be soluble if you combine into the solid. Literally, it can be breakdown into reaction or into process called hydrolysis. That's why there are two methods on how, or uses on how biochemical function of lipid as a basis. The first one is a method based on whether or not lipid can be broken down into smaller units through basic hydrolysis, and that is or that reactions can be water or under basic conditions. So literally that explains that lipids can be or undergo in the process called the hydrolysis. The same with the carbohydrates, because it can broken down smaller pieces, because as we all know, lipids can be soluble if we literally combine it to the solvent. Okay, so that was the methods, or two common methods exist for subclassifying lipids onto families for the purpose of the study. So one method uses is the biochemical function of lipid. Next is based on whether the lipid can be broken down into smaller units through the basis of hydrolysis, or the process by which it has a chemical reaction under the water. Okay. Next, this is a hydrolysis reaction that occurs basic solution in the lipid, and that is called saponification reaction. So basically, if the lipid will undergo with the hydrolysis reaction, it is called as saponification reaction. So basically, the illustration will show you how the saponification occur. So this is constant again on how the triglyceride and sodium hydroxide break down into hydrolysis and reacted to glycerol, and that makes a soap. So that solution is called, or that basic solution is called saponification reaction. Okay, this is literally in the lipids only. Okay. Next is based on biochemical function, lipids are divided into 5 categories. Number 1, energy storage lipids, or that is also known as triacylglycerols, and we have membrane lipids, and that is called phospholipids. As we all know, our cell membrane is composed of phospholipids. Not only phospholipids, but also it is also as sphingoglycolipids and cholesterol. In a biochemical function also, lipid can be emulsification lipids, and that is called bile acid. Lipids can be a messenger lipids, and how come? By the parts of steroid hormones and eicosanoids. And lipids can be protective coating lipids, and how come? By the biological waxes. So this is literally how function, or how can or what are the major function of the lipids. So based on biochemical function, it is divided into 5: energy storage lipid by the means of triacylglycerols, membrane lipids by the means of phospholipids, sphingoglycolipids, and cholesterol; emulsification lipids by the means of bile acid; messenger lipids by the means of steroids, hormones, and eicosanoids; protective coating lipids by the means of biological waxes. So that was the biochemical function of the lipids. So moving forward, based upon whether or not saponification occurs when a lipid is placed in a basic aqueous solution, lipids are divided into 2 categories. So what are those categories? We have saponifiable and nonsaponifiable. So saponifiable is divided also with examples, or with subdivision. In saponifiable, these are triglycerol, glycerophospholipids, sphingospholipids, sphingoglycolipids, and biological waxes. When we say non-saponifiable, these are cholesterol, bile acids, steroid hormone, and eicosanoids. So that's divide or the division of the division of lipids by the means of reaction called saponification. To explain it deeper, let's move forward on defining what is saponifiable lipids and non-saponifiable lipids. So literally, when we say saponifiable lipids, these are converted into smaller molecules when hydrolysis occurs. Okay, when we say saponifiable, it can convert into smaller molecules, and it can convert into smaller molecules by the means of hydrolysis. Not only that, when we say saponifiable, it is a lipid containing long chain fatty acid, which forming one good example of lipid, and that is ester. This lipid have the ability to undergo saponification reaction under the further condition. So that makes saponifiable differ to non-saponifiable. Why? Because non-saponifiable, when we say non, it cannot be broken down into smaller units since they do not react with water. So basically, if we differ the two terminology, saponifiable can convert it into smaller molecules because it can react to the water by the means of hydrolysis, while non-saponifiable cannot be broken down because it cannot react to the water, or it cannot undergo to the process called hydrolysis. So that's differ the two terminologies or the two division of lipids by the means of saponification reaction. So moving forward, our entry point into discussion of these 5 general types of lipids is consideration of molecules called fatty acids. So one good component of lipid is a fatty acid. Aside from the division of lipids in the means of saponification reaction, fatty acid is very important terminologies, or it is a building blocks of the lipids. So when we literally explain fatty acid, these are the structural components of all the lipids that we considered in this chapter, except cholesterol, bile acid, and steroid hormones. With the structural characteristics and physical properties of fatty acid, it makes it easier to understand the behavior of the many fatty acid containing lipids found in the human body. So literally to explain, when we say fatty acid, it is a structural formula also of the lipid. To discuss it more, let's move forward on knowing the types of fatty acid. So fatty acid is a naturally occurring monocarboxylic acid because of the pathway by which they are biosynthesized. Fatty acid is also nearly always contain an even number of carbon atoms and have a carbon chain that is unbranched. In terms of carbon chain length, fatty acids are characterized as, or it can be characterized as what? Long chain fatty acid, medium chain fatty acid, and a short chain fatty acid. So that explains the formula. So going back to fatty acid, if you literally explain what is it, it is, as I've said, it is a building blocks of the fat or the lipid in our bodies and in the food we eat. During actually digestion, the body breaks down a fats into fatty acid, which can then be absorbed into blood, and that makes a fatty acid molecules, which are usually joined together in a groups of 3, forming a molecule called triglyceride. So that explains what is fatty acid all about. And fatty acid can be characterized into 3 chain, and that would be long chain fatty acid, medium chain fatty acid, and short chain fatty acid. So moving forward to explain what is it, so as I've said, one good example or groups that forming fatty acid molecules that explain triglyceride. So as you can see here by the structure of glycerol, we have the long chain fatty acid, another long chain fatty acid, and the medium chain fatty acid. So this is molecular formula, or this is the the particles or the molecules formula in triglyceride explains long chain fatty acid and medium chain fatty acid. So going forward to explain it more, let's go now to the types of the fatty acid, and that was the saturated and unsaturated fatty acid. So the carbon chain of fatty acid may or may not contain carbon-carbon double bonds. On the basis of this consideration, fatty acid are classified into 3. We have saturated fatty acid, or it has abbreviation SFAS. We also have monounsaturated fatty acid, or it has an abbreviation MUFAS, or the last one is the polyunsaturated fatty acid, and it has an abbreviation which is PUFAS, okay, or PUFAS. So that explains how we considered the structural form of fatty acid. Moving forward to for us to know each fatty acid, let's go. Let's discover it or explain it further. So when we say saturated fatty acid, it is a fatty acid which the carbon chain or the all carbon-carbon bonds are single bonds. So the structural formula for the 16 carbon saturated fatty acid is this. So one good example is hexadecanoic acid or palmitic acid, or the common name of it is palmitic acid. As you can literally observe, the bonds is only single bond. Okay, single bond. The only double bond of this is the carboxylic acid or the carboxylic acid. Why? Because as I've said, lipid has its attached on the carboxylic acid. But if you literally observe, it is a component of single bond. When we say saturated fatty acid, single bond. Okay? Next. So the structural formula for fatty acid is usually written in more condensed form than the preceding structural formula. Two alternative structural notation for palmitic acid can be this. So it can be this, which is the branching or the expanded, or it can be a condensed formula, or it can even be a skeletal formula. So this is constant. So I want you to memorize it, familiarize it, because that is one good example of saturated fatty acid. Okay? Moving forward for it. Let's go now to the other fatty acid, and that explains monounsaturated fatty acid. This is a type of fatty acid with a carbon chain in which one carbon-carbon double bond is present. So in biochemical importance of mu-Fas, the configuration about the double bond is nearly always in this section. Let's figure out it in the illustration. So different ways of depicting the structure of mu-Fa as follow. So that explains the monounsaturated fatty acid. Usually the fatty acid or the single bond of it can literally be seen at the center of the structure. So the first of this structure correctly emphasizes that the presence of cis double bond in the carbon chain puts a rigid 30 bend in the chain, such as a bend affects the physical properties of a fatty acid. Why? Because when we literally said monounsaturated, it has a component of cis fatty acid. And when we say cis fatty acid, these are the combination of two carbon, which is bonded together via double bond, and their hydrogen are on the same side of the carbon atom. And that makes or that explains cis fatty acid. So as you considered in the illustration, the double bond is literally at the center of the carbon or at the center of the structure, and you can see that each carbon in the sides has attachment of hydrogen. Okay, so that makes a cis double bond fatty acid or a cis fatty acid that is one component of monounsaturated fatty acid. Okay, moving forward now, let's define what is polyunsaturated. When we say polyunsaturated fatty acid, that explains the carbon chain in which two or more carbon-carbon or bonds are present. So up to 6 double bonds are found in biochemical importance of mu-Fas. So that explains here. So not only one double bond, but it can be 2, 3, or more. So as you can see, in the linoleic acid, we have 2 cis double bonds because we have found 2 double bonds here. 1, 2, and 3. So we have 2 cis double bonds, and we in the simple linear representation of linoleic acid, it can be found as a 2 or more double bonds. Okay, so that's why it's called 2 cis double bonds, because polyunsaturated has already attached with with 2 single bond. Ito na yung sa gitna, kagaya dito sa mono. Sa mono, may isa na siya. So, kapag sinabi natin polyunsaturated, lalagyan na natin agad yan sa gitna ng isang double bond. And, dalawa pang, we will add additional double bond, and that makes 2 cis double bonds or 2 cis fatty acid. Okay? So, that makes the polyunsaturated. Always remember that mono consists to 1 double bond, When we say um, when we say poly, it consists of two or more double bond. And when we say fatty acid, it consists of only only single bond. Sorry. Okay? So that makes the fatty acid differ to monounsaturated or monounsaturated fatty acid and polyunsaturated fatty acid. So moving forward, so these are the example. Good example. For a saturated fat, we have, as you can see, there's no bonded at all. It composed of a single bond, and that explains the fatty acid. And when we say monounsaturated fatty acid, it has a components of one double bond. Okay, as you can see, meron dyan. And when we say a polyunsaturated fat, it consists of double bond. Ito siya. Okay, so that makes a two double bond. So that's why it's called two cis double bond. Kasi dalawa yung double bond na meron siya. Dito sa monounsaturated, it's called cis double bond because we only have one double bond. And when we say saturated fatty acid, that explains single bond. So that is one good examples of the three types of fatty acid, and that is fatty acid itself, monounsaturated fatty acid, and the polyunsaturated fatty acid. Okay, so another examples of selected fatty acid of and in their biological importance. So I want you to familiarize all of that, the name of it and the structure of it. This structure is a skeletal structure. Okay, so I want you to familiarize and memorize this table. Okay, so let's move forward. So the classification or the classify fatty acid with the following structural formula in the ways indicated. So what is the type designation of SFA, MUFA, or PUFA, or this fatty acid? On the basis of carbon chain length and degree of unsaturation, what is the numerical shorthand designation for the fatty acid? And this is actually indication how that the fatty acid can be classified. So from the question itself, it has a solution that what is the type designation of SFAS, MUFA, or PUFA for this fatty acid? So two carbon-carbon double bonds are present in these molecules, which makes it polyunsaturated fatty acid. So literally, those answer or solution by the given question is in at below. You can read it already. Okay? So that is how you classify the fatty acid. So our next topic would be unsaturated fatty acid and double bond position. And we will be classifying the fatty acid on the basis of structural characteristics. And we will be knowing the physical properties of fatty acid. So that explains, or that would be the end of our discussion for this session. So if you have questions, clarifications, you can DM me, or you can PM me, or you can send your question in our GC. So thank you so much for listening. So please watch the video because this is very important to your semi-final examination. That explains all the definition of lipids, the classification of lipids, and the structural forms of lipids, and also the building blocks of fats, which is called fatty acid. So next topic, we will be focusing on unsaturated fatty acid. So if so that's it. That's the end of our session for today. Thank you so much and have a nice day, everyone.